the fever, cold, or sometimes running nose is there. In some of the cases, a high running nose it is detected, whereas in rare cases, the running nose it is detected rarely. Now, the dry cough, it is very, very important, because while coughing, what happens? The cough, it may be mucus type, it may be dry type, but here the infection, when it is caused by means of this coronavirus, it is a dry type. It also causes difficulties in breathing, as well as body ache, sore throat, whereas sneezing from mild to severe, as well as you can find the fatigue also. Now, new coronavirus. Now, the most human coronavirus infection are mild, and the outbreak of this virus was firstly manifested by pneumonia in Wuhan city, the capital of Hubei province in mainland China, with a clinical presentation resembling with viral pneumonia. In human being, generally these viruses set infection either early in the spring or during winter season. After onset of infection, body produces antibody, but they did not last for a longer time, and hence there may be the problem for detection by means of using a rapid test. Sometimes the patient, it may be positive, but while detection, you cannot detect that antibody. That's why there might be some false result is there. Now, depending upon the strains, the antibody, it is produced by the host. Now, antibody for one strain of coronavirus, that may be useless against the other strain of coronavirus. Now, this coronavirus, they belongs from the family Coronaviridae, and the viral particles are polymorphic, which measures from 50 to 200 nanometer, while this new coronavirus, which is responsible to cause COVID, has been reported to be 60 to 140 nanometer in dimension. Whereas these coronavirus are made up of unsegmented, single-stranded, positive sense RNA genome. This genome is contained in a nucleocapsid, which is further enveloped, and this genome, it consists at least 30,000 nucleotides in it, with around 15 genes. This is very, very important. The number of genes, it is only 15. That's why the size of this genome is a spike and projection on its surface, which gives this virus a crown-like appearance, hence the name coronavirus. Now, this virus is having four strains, that is animal, mostly known to infect animals, and the type of coronaviruses which infect to human being, that including human coronavirus 229E, OC43, SARS coronavirus, NL63, HKU1, and MERS-CoV. The natural host of known coronavirus, which is isolated from Wuhan, it was a bat. And from bat, it has been transmitted to the human being. And while this transmission from bat to human being, as the host it is changed, that's why there might be a mutation, little bit mutation in the genome of that virus. That's why from bat, it is surviving into the human being. And now, when it entered into the human being, that have been stabilized the genome. This virus is capable of human-to-human transmission, and this virus, it was found to be 80% similar in its genome sequence to the previously described coronavirus, and has many similarities with SARS coronavirus. Now, the host range for this virus is, it is carried out by bat, sweat, and badgers. Whereas this new coronavirus, that is novel coronavirus in Wuhan, which is similar to SARS coronavirus, which have been isolated and detected during 2003 in Guangdong, which is originated from China, and it has been brought out in winter, which is caused respiratory illness, and traced back to the fresh live animal in an animal market. caused by a mutant strain of already existing coronavirus. Now, the question is raised that whether they are both related with each other, whether the mutation has occurred, so how to have made the new coronavirus jump the natural host via an intermediary to the human being. There are two main transmission methods. The first transmission method, it is by means of direct contact or by droplet-borne transmission. The fact that coronavirus causes pneumonia in human has increased the list of coronavirus causing pneumonia in human being, which is named as HKU1, SARS-CoV, MERS, and novel coronavirus. Now, this mechanism of particle inhibition. Now, basically, what happened when this viruses it is entered through this respiratory tract system, and when it has been entered into the upper part, initially they will set up their infection into the human being. Now, how they will set up their infection? Now, as this virus it is having spikes on its surface, these spikes they are having a receptor, that is, and this surface receptor of epithelial cell of a human being, and the spikes which is having certain protein, they are attached with each other, and after attachment, after specific attachment, then what happened? The upper layer of this virus, that is lipid layer, it is little bit dissolved at the site of attachment, and the nucleic acid it is passed into the host cell. And moment when it is entered into the host cell, it will initiate its replication. I will show you how its replication is initiated. This is one thread I have taken with me. This is, imagine that this is the single stranded RNA of novel coronavirus. This is a positive strand of novel coronavirus. And here I have used the onion. Imagine this onion itself is enzyme which is responsible for the translation of viral RNA. Now, as this is a single stranded positive sense RNA, moment when there is transcription, it is initiated. The transcription it is required for the proliferation or for the replication of the viruses or for reproduction of virus. And for reproduction of virus, as you people may be aware that as they are parasite, moment when they enter into the human host, the whole property of host cell, they will come under the control of viruses. That's why when the property of virus human cell, it comes under the control of viral particle, then what happens? It will immediately initiate to synthesize the various components of these viruses by means of replication of its own genome. Now, as the genome and it becomes positive when it will synthesize all these essential commodities required for this replication of these viruses. For example, the replicase protein, it is also essential. It also needs to synthesize the S protein. It also needs to synthesize the E protein, that is the envelope protein. It also needs to synthesize M protein, that is the membrane protein. It also needs to synthesize N protein required for nucleic acid. It also needs to synthesize the UTR region. And then what happened when this single-stranded genome, it underwent the process of translation by means of this pattern. And when the translation, it progresses one by one, proteins are synthesized. And moment all the proteins are synthesized, now this positive strand, later on that gets converted into negative strand. Then negative strand, one positive strand, it produces varieties of protein. Then later on, parallelly, it will produce negative strand. And after synthesis of a complete essential requirement, then the negative strand once gets converted into positive strand. Moment when it becomes a positive strand again, then it will enter into the next cycle of its replication, and it will initiate with translation by means of a suitable enzyme, that is the RNA-dependent RNA polymerase, that is RDRP. This is an enzyme, RNA-dependent RNA polymerase, whereas in majorities of microorganisms, where the DNA as a nucleic acid, where the DNA polymerase enzyme is required. Where this DNA polymerase are of again three types: DNA polymerase 1, DNA pol 2, pol 3. But here such types are not there. Only RNA-dependent RNA polymerase enzyme is here, and this RNA-dependent RNA polymerase, it synthesizes this new RNA molecule. Now, after this synthesis, when the particle synthesize two particles, then continuously the replication of viruses, it is progressive or continue. And after continuation of replication, it will affect one by one cell. And moment at that time when it is affected, at the same time our body, it will produce a certain chemical. Now this mechanism is very, very important. Basically, I would like to focus on this slide. Moment when the cell it infects the human host, the human cell it is affected, the Lord has given a very big mechanism to our host, that is, it will synthesize and naturally certain chemicals, which is named as a cytokine. Basically, in our body, all specialist doctors are available. Please trace on my this sentence. Our body, it is governed by all specialists of doctors. Moment when there is an attack of virus, our infected cell itself produces certain chemical, and that chemical it is named as cytokine. And among this cytokine, the most important cytokine, it is interferon. And this interferon, it is produced by means of our body or our cell or infected human cell, and this is sufficient interferon only it is produced when the human host is healthy. That's why the government of India, not only government of India, but all government from all over the world, they